Hello, students. I hope everyone is doing all right today. So we are going to begin the last two sections of 10.3 speciation. We're going to look at speciation first and what that means. So you probably recall this term from ninth grade bio. And here in the essential understanding, it pretty much gives you the cause of speciation. So why does speciation happen? So speciation is due to divergence of isolated populations. And we know that this divergence of two separate species can be gradual, or sometimes the pace can be a little bit faster. So if you share a common ancestor like so, and here you have species 1, so that species 1 can continue to diverge from perhaps a species 2. So we know this as gradualism. But we're also going to look at a different pace of speciation in our next section called punctuated equilibrium. So we know that speciation is an evolutionary process that results in the formation of a new species from a pre-existing species. It occurs when there is some type of isolating mechanism. So it's not always a reproductive isolating mechanism, but that means that now these two breeding organisms cannot produce fertile, viable offspring. And little by little, they start becoming different species. So there are two basic mechanisms by which speciation can occur, and we're going to be looking at those today. So allopatric speciation is when there's a geographical isolation due to some type of geographical barrier, and sympatric speciation is due to some form of reproductive isolation. So let's look at the examples right now. So just like I said, in allopatric speciation, there's some form of geographical barrier, and this isolates populations from the ancestral species. So the population that is separated, as shown here in this diagram, it begins to evolve separate. So now you have two populations here that begin to evolve separately, and all of this is due to mutations, genetic drift, and natural selection. And they're going to get to a point where they're so different that they can no longer interbreed. Okay, so that's allopatric. In sympatric speciation, there is no physical barrier. So the two different species that arose from a common ancestor, they are in the same geographical location, but now you have some type of reproductive isolation. So then that means that the new organisms, which are now considered a new species, they have some form of genetic abnormalities or some form of chromosomal error. And that means that they cannot interbreed with the original parent population. So this would be the sympatric speciation. So in the application here, we're going to see the speciation in the genus Allium, which is a genus for different forms of onion. And we're going to see how the genus Allium can form these different species by a process known as polyploidy. So we're going to look at what that process is in just a second. So just like I just mentioned, sympatric speciation is when you have some type of reproductive isolation due to some type of genetic abnormality. So the most common cause of this genetic abnormality is meiotic failure during gamete formation. So then that means that this parent cell right here is, when it undergoes cytokinesis in meiosis, it's going to fail to properly separate the chromosomes. So let's say that this parent is originally diploid and there's some form of meiotic error. So by the time meiosis 2 is finished, you're going to see that the gametes here are also diploid. So the chromosomes did not separate properly. So having additional sets here of chromosomes that are, they could be similar to the parent, or they can even be more than the parent, is a form of polyploidy. So you end up with additional sets of chromosomes. So then what happens? Well, these gametes can self-fertilize, right, if it's a plant, and then that means that you're going to get 2N plus 2N, and you're going to end up with a tetraploid or 12 chromosomes here. And this tetraploid, or this, which is a form of polyploidy, they can be viable organisms. They can be fertile organisms. So you can have one tetraploid here breed with another tetraploid, but they just can't breed with the original parent population. So that's why we consider these polyploids a different species altogether. Now, fertile polyploid will typically require 2 polyploid parents. So we just saw this here, right? A 2N plus a 2N giving you a tetraploid. But there could be some forms of allopolyploidy. And this is when you have perhaps a diploid gamete and a haploid gamete. And then you're going to end up with an uneven number of chromosomes here. So you're going to end up with an infertile triploid zygote. So anytime that you have this uneven number here, then that means that the offspring is going to be infertile. So usually, right, if it's an even number, they're fertile, but if they're an uneven number, they're infertile. All right, so why is polyploidy a desirable trait for farmers? So here we see that some forms of polyploids, for example, the triploid watermelon, since it is infertile because it has an uneven number of chromosomes, it's not going to produce any seeds. And, you know, sometimes that is favorable because the consumer likes that. And then typically polyploid crops are going to grow larger, and they're going to demonstrate improved longevity and disease resistance. And this is what we refer to as hybrid vigor. So then farmers may induce polyploidy in certain plants by treating plants with certain drugs known as colchicine. And with this type of drug, what you're doing is that you are aiming to produce some type of meiotic error. Okay, so you want the mistake to happen. So in this type of hybrid vigor example, here we see a normal onion, right, diploid, and this is the normal onion plant, and this is what the normal onion looks like. But if then you get a polyploid plant, so you're inducing this plant to become polyploid, meaning that you take 2 diploid gametes and you fertilize them to get a tetraploid here, then this plant is going to be a polyploid. This plant is going to be a tetraploid. So this plant is going to be much larger than the normal onion. So this is, again, what we refer to as hybrid vigor in polyploids. So here you see speciation in Allium. And just like I said before, the genus Allium is comprised of monocots, and they include onions, garlic, chives, and leeks. And you see different forms of polyploidy here. So here you see diploid, tetraploid, and octoploid. And these are all forms of fertile Allium species. And then we see the triploid and the pentaploid, and these are unfertile form of Allium species. So here they're all different examples of polyploidy. All right, so that's it for today.